Hi, this is Emperor Nadu. Today we will discuss about the central molecular biology, like central dogma of molecular biology. Then this central molecular biology of central dogma of molecular biology is like replication, transcription, translation. So replication may make a DNA copying, like DNA to DNA synthesis, like it is replica, that is making of DNA to DNA, and transcription, DNA being made into the RNA, that means DNA conversion, like DNA synthesis of RNA. What are the translation? Translation is RNA synthesis of proteins. These are the various mechanisms. Also post translation modification also is there. Replication, remember this is the replication is simultaneously both it is bidirectional replication and DNA to DNA synthesis, like some of the things will be reverse transcription, maybe occur DNA to RNA to DNA synthesis. So like, but here it's semi-conservative model. There are the conservative, semi-conservative models also is there. So, but coming to this, we are talking about the semi-conservative. Replication is one is 5' to 3' and 3' to 5' directions. Replication always will be occur 3' to 5' only. So how the replication is semi-conservative, heavy, hybrid and light chains. So replication role enzymes, topoisomers, and these what are the requirements, like replicase, helicase, gyrase, ligase, and also DNA polymerase, like DNA polymerase 1, 2, 3, and DNA binding proteins like ligands, leading strand and lagging strand. So this is the leading strand and this is the lagging strand. Here is some of the small thing, RNA primers and DNA polymerase here. So what are DNA ligase? Small leaks will fill up here. This is the helica. So unwinding process will be going on there. So this is topoisomerase. This is halo enzymes. Also single stranded binding DNA polymerase. Replicase helix opan causes like helicase and causes the supercardying and DNA binding proteins. Also replication leading strand 3' ends and of template, and as opens support the DNA polymerase binds makes a new DNA 5' to 3', same directional opening helix made continuously. So how the continuously opening, like this is small as the DNA single strand proteins will be binding here. So after unwinding to inhibit the rewinding process. Replication lagging strand, a 5' and cannot be continuously. So direction will be RNA primers, new DNA made 5' to 3' opposite directionally is discontinuous. So here we can see about the Okazaki fragments about in these things. Transcription is the RNA DNA to synthesis of RNA, and one DNA strand is template, and the other just for replication antisense. So in nucleoli from following DNA strands determines RNA sequence, like 5', 3' to 5'. So here is how transcription is stable. Here is how it will be conversions of DNA to RNA. So transcription DNA is opens up enzymes like RNA polymerase, like binds with which strand. So using the template strand is ROT 5' to 3' is called heterogeneous nucleus RNA. How does RNA polymerase know where starts? Upstream promoter sequence and prenome box and data box. RNA polymerase starts with the transcription X nucleotides downstream of data box. Introns and extrons. Introns intervening sequence not all DNA codes for the proteins. Regularly inform junk for DNA. Exons codes for proteins. So processing of heterogeneous nuclear RNA into the mRNA. 3 steps introns of remote self splicing, 5' methyl guanosine cap added and polyatyl added and moved to cytosol for translation. So this is how heterogeneous nuclei into the mRNA process. Translation RNA to protein and change from the nucleotide language to amino acid language on ribose. So vectorial nature is preserved. So 5' end mRNA becomes amino terminus of proteins. Translation depends on genetic code. Today we are discuss about the central dogma of the molecular biology. So what are the replication, transcription and translation? These are the process where the DNA to RNA, RNA to proteins of synthesis. DNA to DNA is replication, DNA to RNA is protein, and RNA to protein is RNA to protein, like this is translation, and DNA to RNA is transcription, and also DNA to DNA is replication. Some of the processes like DNA to RNA to DNA, that is reverse transcription, also is there some clinical significances. Thank you very much.